Bio-Technology. Now, here what are the remaining options? The eugenics. What is meant by eugenics? Applications of genetics for the welfare of human being society. Application of genetic principles, that we call it as eugenics. What is meant by gene slicing? That means to remove the unwanted genes. For example, you know very well, gene regulation and expression in eukaryotes. In eukaryotes DNA, exon introns are present, where the introns are not useful for the translation. They are removed. That we call it as slicing. Now, the next one. Hybridoma technology was invented by Georges Kohler and Cecil Mainstein. Now, the hybridoma technology was introduced by Georges Kohler and Cecil Mainstein. Now, what is meant by hybridoma technology? Now, from the rat body, they collected the cancer cell. It is also called the myeloma cell. What is the property of the cancer cell? It can divide. Now, in the rat body, from the spleen, they collected B cell. That is called the B lymphocyte. The B lymphocyte produces antibodies. The B lymphocyte cannot divide but produces antibodies, whereas the cancer cell can divide but do not produce antibodies. Now, what they did? The B lymphocyte crossed with the cancer cell. They are getting hybridoma cell. Now, you have to imagine what are the properties of hybridoma cell? Like that of the B lymphocyte, it can produce antibodies. Like that of the cancer cell, it can divide. So, it is having the properties of both the cells. This hybridoma cell is used in various purposes. Such hybridoma technology first invented by Georges Kohler and Cecil Mainstein. For that, they got the Nobel Prize. Now, what is the answer? The answer is the Georges Kohler and Cecil Mainstein. Here, as we observe the Watson and Crick, they discovered the molecular structure of DNA. They explained the central dogma of molecular biology. We already discussed it in the genetics. What is meant by central dogma of molecular biology? The DNA transforms its biological information to the protein molecule through mRNA. That is the central dogma of molecular biology. Now, the next question. In recombinant DNA technology, the DNA is cut into fragments at specific sites by using. In the recombinant DNA technology, the DNA is cut into specific sites. Now, this is the DNA strand 5' 3' 5' 3'. This DNA strand can cut at specific sites. That means in a palindrome sequence. The DNA strand can cut at specific sites by using what type of scissors? Now, here one is ligases, second one liases, third one exonucleases. What about the restriction endonucleases? Now, as we observe the exonucleases cut the DNA strand terminally. Okay, you observe the diagram. Exonucleases cut the DNA strand externally. What about the endonucleases? They cut the DNA strand internally. But what about the restriction endonuclease enzymes? They cut the DNA strand at specific sites. That means in a palindrome sequence. So, here What is the reason for the popularity of the recombinant DNA technology? That is the invention of restriction endonucleases. Now the answer is, now the next question. In recombinant DNA technology, the fragments formed by using restriction endonucleases are separated. Now it is a DNA strand. Now the DNA strand is taken in a petri dish or in a beaker. Now this is the DNA strand. Now we use restriction endonuclease. The restriction endonuclease will cut or cleave the DNA strand in a palindrome sequence. After completing the process, the fragments are separated, smaller fragments and larger fragments. In smaller fragments, less number of base pairs. In larger fragments, more number of base pairs. The restriction endonucleases will cut the DNA strand in a palindrome sequence. Their work is over. Now the fragments are separated by what type of process? That we call it as gel electrophoresis. For example, this is the DNA strand. Now the restriction endonuclease, it cut the DNA strand in a palindrome sequence. By gel electrophoresis, the fragments are separated. The process of separation of fragments after completing the work by restriction endonuclease enzymes, that we call it as the gel electrophoresis. Now the answer is, the next question. Most commonly used cloning vector, generally for carrying the gene. Gene means part of DNA. For carrying the gene, we use different vehicles. Here the vehicles are called vectors. What are the vectors? The plasmid, bacteriophage, cosmid, bacterial artificial chromosome, yeast artificial chromosome. All these are the vectors. What is the most popular? Plasmid. That plasmid, so many varieties are present. The most popular, PBr 322. Here the P means plasmid. Already in the last class we discussed. Here the B means Bolivar. And R means the Roderick. The number of this vehicle is 322. So many plasmids are present, but PBr 322, that is the most popular vector. Along with the plasmid, we can introduce only small fragment of DNA. Now here, most commonly used cloning vectors are plasmids, phasmids, cosmids and slimeouts. Now the answer is, next question. Small extra chromosomal double-stranded circular DNA molecules are bacteria used as cloning vectors. Now where the plasmids are present? Now this one we call it as the bacterium cell. This is bacterium cell. This is cell membrane. And this is the cytoplasm. Now we know very well the bacteria are the prokaryotes. Here distinct nuclear membrane absent. Now this is the cytoplasm. Actually here what is present? The nuclear material, but no distinct nuclear membrane. Outside nuclear material in the cytoplasm, now the double circular, what is present? The plasmid. What is meant by plasmid? It is DNA, a type of DNA present outside the nuclear material in the cytoplasm. It is called extra chromosomal DNA. Now based on this, what is the answer is? The answer is the plasmid. The next question. Bacterial cells are treated for extraction of plasmids Now the plasmid is present inside the bacterium cell. Around the bacterium cell, what is the present cell membrane? How we have to remove the plasmid? The bacterium cell is dissolved. In order to dissolve the bacterium cell, what is used? The lysozyme or EDTA solution. Now here the answer is bacterial cells are treated for extraction of plasmids by using isozyme, lysozyme, lipase, trypsin. The answer is the lysozyme. The next one. In recombinant DNA technology, the restriction endonuclease is used cleaving, that means to cut. Cleaving the donor DNA is also used to cleave the vector DNA. Now in the recombinant DNA technology, we take the donor DNA. The donor DNA is cut by using restriction endonuclease enzyme. In the same way, the donor DNA is carried by vector. We have to use regarding the vectors, it may be plasmid, it may be bacteriophage, it may be cosmid, or it may be bac, or it may be yak. Now, vector, it is nothing but a DNA. Now, with the help of restriction endonuclease enzyme, the vector also cleaved. That means the donor DNA is complementary to that of the vector. That means this is the donor DNA. From the donor DNA, a small fragment is removed. Now, this is vector. From the vector, a small fragment is removed. From the donor DNA, a small fragment is removed and that is attached to vector. Now, this donor fragment is complementary to that of the vector. Now, both are united. We are getting the rDNA. Now, here, in recombinant DNA technology, the restriction endonuclease used cleaving the donor DNA is also used to cleave. You have to check the word. Is used to cleave the vector DNA. So the statement is correct. The ends of cleaved DNA vector must be complementary to the ends of cleaved donor DNA to form the chimeric DNA. Donor DNA, vector DNA, both are united. We called recombinant DNA. This recombinant DNA having the properties of vector as well as the properties of donor DNA. This is called chimeric DNA. Chimera, that is a type of fish. It has the properties of cartilage fishes as well as the bony fishes. So that here the recombinant DNA having the properties of donor DNA, vector DNA. So that we use the word chimeric. Now the answer is both are correct or he explains here. The next one. Arrange the following events in recombinant DNA technology to get the correct procedure of the technique. Now as we observe the protocol of recombinant DNA technology, first of all we have to select or isolate the donor DNA. Now from the donor DNA a small fragment is removed. Now we are getting a fragment from the donor DNA. In order to carry this donor DNA, we select one vector. The most popular vector is plasmid. From the vector, a small fragment can be removed. Now, the fragment of donor DNA attached to the vector, we are getting recombinant DNA. It is also called the chimeric DNA. Now, we want to get more and more copies of the donor DNA. Now, this entire particle is kept in a bacterium cell. Now the bacterium cell is kept in the culture. After completing the culture, we have to isolate the DNA which is cloned. How we have to collect with the help of genetic markers or colony hybridization. Later we have to develop. Now, arrange the following events. Arrange the following events in recombinant DNA technology to get the correct procedure of the technique. A, B, C, D, E, F. Now what is the first one? Cleaving the donor DNA at specific sites using restriction endonucleases. Now the fragments are separated by gel electrophoresis. Now we go for the B. Separation of specific DNA fragments by gel electrophoresis. Then we go for the vector. Now transfer of specific donor DNA to cleaved vector DNA to form recombinant DNA or chimeric DNA. Then we go for transfer of chimeric DNA into suitable recipient bacteria. Then we have to identify. How to identify? The selection and collection of transferred bacteria with the help of genetic markers or colony hybridization. Then we increase its number, that is proliferating the transferred bacteria. Now you check the option, the third one. Again you have to observe the option. Now we go for the next question. Introduction of chimeric DNA. What is meant by chimeric DNA? The part of donor DNA and the part of vector. Now we are getting the recombinant DNA. This recombinant DNA has the properties of vector as well as donor. So it is called recombinant DNA or chimeric DNA. Now, introduction of chimeric, just you refer the word, there is a small mistake, chimeric. Introduction of chimeric DNA directly into the recipient bacterial cell by using a virus. With the help of virus, we are introducing the chimeric DNA into the bacterium. With the help of virus. By using the virus, we are introducing the chimeric DNA into the bacterium cell. That process we call it as the transduction. The answer is. Now the next question. Donor DNA fragments is joined to complementary ENSA vector DNA by using. From the donor DNA, we are taking one fragment with the help of scissors. Here scissors means enzymes. From the donor DNA, a small fragment is removed by using restriction endonuclease. Now thus that fragment of donor DNA attached to the vector. How the two strands of DNA are attached? With the help of an enzyme called ligase. Now again repeating, exonucleases, they are also useful to cut the DNA. Endonucleases, they are also useful to cut the DNA. Restriction endonucleases, they also cut the DNA in a palindrome sequence. The two DNA strands or fragments are attached by a glue or an enzyme that we call it as ligase. Now the answer is, the two fragments of DNA attached by an enzyme called the ligase. The next one. Read the following and find the correct match of recombinant DNA technology. So far we discussed some points regarding the recombinant DNA technology. Based on that, you have to choose the correct matching. The first one, EDTA. The EDTA is used to dissolve the bacterium cell wall. Ligase, it is used to attach the two fragments of DNA. Restriction endonuclease, it cut the DNA at palindrome sequence. Because of the restriction endonuclease enzyme, we are getting the sticky strands. Whenever we are going to get the sticky strands, the restriction endonuclease enzymes cleave the DNA in a palindrome sequence. So we are getting the sticky strands. Now the colony hybridization. After completing the culture in the laboratory, we have to identify the cloning DNA with the help of genetic markers or colony hybridization. And now, based on this information, you have to observe the answer in a fraction of second. The answer is the first one. Now the next question. This is used a molecular gum. Gum means to attach or to glue. In the recombinant DNA technology, the fragment of donor DNA attached to the vector DNA. Here the two fragments are attached by a glue. Here what is used for the glue? Enzyme. Here what is the enzyme? The ligase. Now here the answer is the DNA ligase. The next question. Cosmid is a hybrid. Now we are having an idea regarding the vectors. One is phasmid. Along with the phasmid, we can introduce only small part of DNA but not entire strand of the DNA. Whatever the bacteriophage, along with the bacteriophage, we also introduce only small fragment of the DNA but not entire strand of DNA. Now, phasmid, plasmid and bacteriophage, these two are united by Cos technique. We are getting cosmid. With the help of cosmid, with the help of bac, with the help of yak, we can incorporate the entire length of DNA strand. The length of DNA strand is 2 meters. Entire length of DNA strand can be incorporated by using the cosmid, by using the bac, by using the yak. Now, assertion. Cosmid is a hybrid vector. How it is obtained? By fusing the plasmid and bacteriophage. So cosmid is a hybrid. It is powerful than that of the parents. Reason, cosmid is produced for specific purposing by joining the cos site of phages and plasmids of bacteria. So the answer is both are correct or he explains here. The next one. Cosmids are used as vectors for incorporating relatively longer foreign DNA into recipient cell. Along with the plasmid, only small fragment of DNA. Along with the bacteriophage, only small fragment of DNA. But by using the cosmid, bac and yak, we can incorporating the entire length of DNA. Because the reason, cos sites of cosmid facilitate the incorporation of, that means insertion of, relatively longer foreign DNA fragments into recipient cells. Here what are the recipient cells? The bacterium cell. And plasmid sequence is useful in the replication. Now the answer is both are correct or he explains here. Now the next one. Among the following is useful for transfer of a relatively longer DNA fragment into the recipient cells. One is cosmid. Second one bac, bacterial artificial chromosome. Yak, yeast artificial chromosome. By using these three vectors, we can introduce the entire length of the DNA strand. So, you imagine the answer. Now the next question. if the sequence of bases on one sticky end of DNA fragment is, now here, if the sequence of bases on one sticky end of DNA fragment, that is 5 dash. Now here A, T, T, G, G, A, T. Now you read the information in the forward direction. Now what is the complementary? A, T, C, C, A, T, 3 dash. Because for this it is the complementary. And now what is the answer for this question? The third one, 3 dash T, A, C, C, T, A. That means it is complementary to this one. Now the next one. Read the following statements. Read the following statements about recombinant DNA technology and check the incorrect. We are asking incorrect statement. Now the first one. Specific proteins which are useful as molecular scissors. What are the molecular scissors? Enzymes. Enzymes are made by proteins. Proteins are made by amino acids. That is the concept. Specific proteins which are useful as molecular scissors are the restriction endonuclease enzymes, because these are the enzymes made by proteins, they are made by amino acids. So this is the correct. Second, if a restriction endonuclease cut the DNA at the center of symmetry, DNA fragment produced are with blunt ends. Now this is the DNA strand. 5 dash, 3 dash, 5 dash, 3 dash. Now this is the center of the DNA strand. That is symmetry. Okay. If the enzyme cut the DNA strand at symmetry, we are getting the blunt ends. These are called the blunt ends. But as we observe the restriction endonuclease enzymes, this is G, A, T, T, C. Okay, this is one strand. And this is another strand. Now what is the complementary for this? G, A, T, T, C. Okay. Restriction endonuclease enzyme cut the DNA strand in a palindrome sequence. Now as we observe, now this is sticky ends. Here we use a new fragment. Here we use a new fragment. Because of the restriction endonuclease enzymes, cut the DNA strand around symmetry but not at symmetry. If they cut at symmetry, we are getting the blunt ends. Okay. Around symmetry, we are getting the sticky ends. At symmetry, we are getting the blunt ends. So what type of ends are useful in the recombinant DNA technology? Sticky ends. What type of enzymes cut the DNA strand by getting the sticky ends? The restriction endonuclease enzymes. Now, the second one is correct. Third, if a restriction endonuclease cuts the DNA around the line of symmetry, the DNA fragments are formed with cohesion ends. They are also called the sticky ends. What about the fourth one? The selected DNA fragment is incorporated into the vector DNA with the help of the PEG. No, with the help of the virus or other methods, but not the PEG. So, the incorrect one here, The given answer is the third, that is the wrong. The fourth one is the wrong. Please you check the answer. Here in the given 4 statements, first 3 are correct. The last one is incorrect. The last one is incorrect, but not the third. Please check the answer. Arrange the following events of PCR, that means polymer chain reaction. Arrange the following events of PCR in a proper order. Now, this is the DNA strand. 5 3 5 3 Now they are connected by the hydrogen bonds, we know very well. In the PCR, the first one is, we know very well, denaturation, followed by annealing, followed by extension. Now here the temperature 94 degrees centigrade. Here the temperature is, that is 54 degrees centigrade. Here the temperature is 72 degrees centigrade. For this 1 minute is required. For this 45 seconds. For this 2 minutes. Now, because of the denaturation, we are increasing the temperature. The 2 strands are separated. The 2 strands are separated. Now each strand acts as a template. Now we use the probes. What is meant by probe? A single strand. Now these probes exhibit the movement between 2 strands until the complementary is available. The probes exhibit the movement between the 2 strands until the complementary is available. Such movement we call it as the Brownian movement. Whenever the probe gets the complementary information, automatically that is attached. The probe attaches at 3 The probe attaches at 3 and extending from 5 to 3 Now you have to observe the first statement, denaturation of double-stranded DNA fragment into single-stranded pieces. This is the double-stranded. In the denaturation, both are separated. Both one act as a template. We are not discussing the protein synthesis. We are discussing the PCR. Both the strands act as the template. Now, attachment of primers to the DNA strand. Isolation of desired DNA for the cloning. Attachment of DNTPS to the template strand. Now, you observe the answer. Now, the first one is the isolation of desired DNA. Now, this is the desired DNA. Now, we go for denaturation. In the denaturation, the 2 strands are separated. Now, both are separated, each one acts as a template. Now, the next one is attachment of primers. Here the primers means the probes. Attachment of primers to DNA strand. Now the last one is attachment of DNTPS. What is the function of DNTPS? They extending the length of DNA strand. The DNTPS extending from 5 to 3 We are reading the template from 3 to 5 We have to observe such type of question in the next one. Now, read the following statements regarding the PCR and note the correct. The primers are attached to a single strand of DNA with the help of DNA polymerase. We are using the RNA polymerase. Second one, bonding of primer to single strand of DNA is only at the 5 no, at 3 The third one, NTPS are added from 5 to 3 and we read the template from 3 dash to 5 dash. So the third one is correct. Fourth, annealing. Annealing is done at 54 degrees centigrade for how much time? The 45 seconds. Now you observe the answer. Third statement, fourth statement are correct. First one, why it is wrong? We are using the RNA polymerase. And next, second one, not at 5 dash, at 3 dash. Next question. In PCR, extension of DNA chain, first one is denaturation, second one annealing, third one extension. It is done at what temperature? 72 degrees centigrade temperature. The time required is 2 minutes. It is the ideal temperature. At this temperature, the extension is going on. Now, A, in PCR, extension of DNA chain is performed at which temperature? 72 degrees centigrade. For how much time? 2 minutes. Now, this is the ideal working temperature for PCR is 72 degrees. That means ideal means it is favorable temperature. It is the suitable temperature. At this temperature, the DNA strand extends. Now the answer is the next one. Miniature chemical factories. In the biotechnology, in the industries, we have to use what type of organisms? The microorganisms. So here, miniature chemical factories. Miniature, that means they are very, very small. In the biotechnology, in the industries, we have to use microorganisms. They are called miniature. So the answer is the microorganisms. The next question. Secondary only metabolites. Now, by using the microorganisms, we are getting two types of products. One we call it as vitamins. The vitamins not only used by us, but also used by microorganisms. The second one is antibiotics. Antibiotics are used by us. They are not useful for the microorganisms. So here, the secondary only metabolites that are antibiotics, they are used by us but not the microorganisms. The next one, products in microorganisms, that means products obtained from the microorganisms used by themselves and also used by us. From the microorganisms we are getting the vitamins, vitamin B2, vitamin B12. These vitamins not only used by us but also used by the microorganisms themselves also. Now the answer is primary metabolic products. Here what is the meaning of primary metabolic products? Vitamins. Now the next one, products obtained from the microorganisms which are not used by them but are used by us. From the microorganisms we are getting antibiotics, erythromycin, penicillin, gentamicin. All these are the antibiotics. These antibiotics are used by us to kill the microorganisms. So antibiotics are not useful for the microorganisms. They are used by us to kill the microorganisms. So the answer is the secondary metabolic products. What are the secondary metabolic products? Antibiotics. Now the next one, the fermentation product of sugars is converted to acidic acid by yeast. For the preparation of acidic acid, we have to use what type of microorganism? Here we are asking yeast. What type of yeast is used in the preparation of acidic acid from the sugars? Saccharomyces cerevisiae. If it is bacterium, the answer is Acidobacter. But here we are asking the yeast. In order to prepare acidic acid from the sugar, we use the yeast Saccharomyces. If it is a bacterium, the answer is Acidobacter. Students, in the next class, we are discussing the remaining part of biotechnology, and in the last class, we have to discuss the last topic of the second year.